And the mission today is really all about synthesis. It's about getting profound insight into structure. Our source material is, well, it's a comprehensive look at how cells integrate to form tissues. We'll be focusing specifically on the four primary tissue categories: epithelium, connective tissue, nervous tissue, and muscle tissue.
Okay, so all four. That's a huge landscape.
It is. But while those last two, nervous and muscle, are absolutely crucial for, communication and movement, we are going to dive deep, and I mean heavily, into the foundational architecture. So that's the epithelium and the connective tissue. The goal is to thoroughly grasp the classification, the structure, and the critical function of these basic building blocks that make complex multi-system life even possible.
Right, and this isn't just academic, is it? This is the fundamental how of your body. It's about understanding the specific cellular engineering that gives your skin its incredible barrier function, that provides your tendons with unbelievable tensile strength, and that allows your kidneys to filter your blood with microscopic precision. Prepare for a masterclass in biological architecture as we unpack cellular society.
Let's do it.
So when we usually talk about life, the starting point is always the cell, right? The fundamental unit.
It is. But the thing is, in a multicellular organism like us, the vast majority of cells rarely exist as single free-living entities. Instead, they cluster together into what we call tissues. Tissues are essentially dedicated teams of cells. They specialize during development to carry out these highly specific, coordinated functions.
That initial specialization is just remarkable to me. The source notes that the human body has more than 200 different cell types. Yet every single one of them, every one, carries the exact same complete genome. They all start with the same blueprint. So how is it possible for them to end up as something as wildly different as a bone cell versus a nerve cell?
It all comes down to gene expression. It's about selectively reading that massive, massive blueprint. Out of the, potentially tens of thousands of genes available, each cell type expresses only a very specific subset of that genetic program.
So they're only reading certain chapters of the book.
Exactly. And this selective expression is what drives them to specialize, both morphologically in their shape and functionally. You get these dedicated teams. Epithelial cells specializing in protection and transport, muscle cells in contraction, nerve cells in signaling, and so on. It is the ultimate division of labor at the cellular level.
Okay, so that initial specialization sets the stage. And once they're specialized, these 200-plus cell types fall pretty neatly into just four major categories, which, as you said, is our roadmap for today.
Exactly. These four categories are defined by their structure, their primary function, and their specific molecular properties. They are epithelium, which is for covering and lining, then connective or supporting tissue for structure and transport. Then you have muscle tissue for movement, and finally nervous tissue for communication and control. And these four tissue types, they interlock, they communicate, and they form all the organs and systems in the body.
And for this deep dive, we're really focusing on those structural components, the connective and epithelial tissues. They form the mechanical apparatus and the defensive layers, right?
Yeah, they do. And this specialization, while it enables all this incredible complexity, it also creates an enormous vulnerability. The implication is profound. If the integrity of any of these specialized systems fails, a disease state is almost immediately supported.
So a failure in one small part can cascade through the whole system.
Instantly. For example, if the delicate balance of the connective tissue, the stuff providing the structural framework, is compromised, or if that protective layer of the epithelium loses its barrier function, the entire system is at risk. A tiny failure in the specialized structure of something like type 5e collagen, which acts as the foundation beneath the epithelium, can rapidly lead to catastrophic renal failure. It just shows how dependent the body is on the perfect integration of these foundational tissues.
Okay, so let's start with that absolute foundation then, the epithelium. When we talk about surfaces in the body, we're really talking about epithelia. Where exactly do we find it?
Epithelium is the boundary layer. It performs three defining architectural roles. First, it covers all of your body surfaces, like your skin. Second, it lines every internal body cavity, illuminating your gut, your blood vessels, your kidney tubules, all of it. And third, it forms all of your glandular tissue. Its defining feature is that it separates the internal environment from the external world, or it delineates internal compartments from each other.
You mentioned it has three crucial roles, the three E's, I think you call them. Let's break those down.
Right. They define its primary function. The first is protection. Epithelia act as this continuous physical barrier against, abrasion, chemical insults, heat, microbial pathogens.
The first line of defense.
Absolutely. The second is the selective barrier function. This is more active. The epithelium is actually managing the passage of substances. Think of the kidney deciding what to filter out of the blood or the gut selecting which nutrients to absorb. This highly controlled passage is entirely dependent on that epithelial layer.
So it's a gatekeeper.
Precisely. And the third is secretion and absorption. Epithelia form the functional surfaces for glands to export chemicals, for the lungs to exchange gases, and for the intestines to pull in nutrients.
That structural role as a barrier must require extreme cellular organization. Are the cells just, stacked up like bricks, or is there a specific method of adhesion that holds them all together?
The organization is absolutely the key. What's so fascinating here is that epithelia are almost entirely cellular. They're known for having minimal extracellular material between the cells. Unlike connective tissue, which is matrix heavy, here the cells themselves form the structure. And they are bound together in these tight sheets by complex junctional complexes.
Like microscopic zippers and rivets holding them all together.
That is a perfect way to put it. You have three main types working in concert. The tight junctions, or zonula occludens, are exactly like the zipper. They form a continuous, impermeable seal around the top surface of the cells, strictly regulating what can pass between them.
So nothing slips through the cracks.
Nothing gets through without permission. Below that, you have adherent junctions, which anchor the cell's internal skeleton, and then you have desmosomes. Those are the physical rivets. They provide immense spot-weld mechanical strength to resist physical shearing forces. These junctions are what control the critical permeability of the layer and give it its mechanical strength.
Okay, so if the cells are packed that tightly together and they're forming this boundary layer, they must be cut off from the main blood supply. How do they survive? How do they get nutrients and oxygen?
This is a critical point. Epithelia are avascular. They lack blood vessels entirely. Therefore, they have to rely on diffusion for all of their nourishment, their oxygen, and their waste removal. This all has to occur through the basal lamina, that foundational layer from blood vessels located in the underlying connective tissue.
So that basal lamina has to be incredibly thin and efficient.
It has to be, yes. This arrangement necessitates a very efficient, very thin foundational layer to allow for this passive molecular exchange across the barrier.
And given that they are the boundary layer, they're taking the brunt of every mechanical or chemical insult. Their ability to repair itself must be exceptional.
It is essential for survival. It has to be. Epithelia possess an exceptionally high regenerative power. They can rapidly replace lost or damaged cells. This process is driven by mitotic cell divisions that take place primarily in the basal layer, which is closest to that nutrient source in the connective tissue. New cells are produced at the base, and then they migrate upward to replace the cells that are lost from the surface. You can think of it as a constant conveyor belt of renewal. It's crucial for wound healing and for maintaining the integrity of surfaces like your skin and the lining of your gut.
And does that change over time?
It does. We do see a decrease in this regenerative capacity as we age, which is one of the main reasons why wound healing tends to slow down later in life.
Okay, so let's unpack the architectural diversity here. The source says epithelia are first categorized by two really simple questions. How many layers are there, simple or stratified? And what is the shape of the cells on the surface, squamous, cuboidal, or columnar? And this categorization immediately tells us what the tissue's priority is: speed, capacity, or protection.
That's it, exactly. We can start with the unilaminar, or simple epithelium, just a single layer of cells. The single layer structure is the optimal design when the priority is efficiency and rapid transport. So filtration, absorption, or secretion. If the cell's main job is passive exchange, you want the absolute shortest possible distance for molecules to travel.
So if the priority is pure speed, we use simple squamous, flat cells.
Precisely. Simple squamous cells are flat, thin, and tightly packed. They're often compared to pavement stones or a thin sheet of cellophane. Their cytoplasm is what we call highly attenuated. It's stretched incredibly thinly, which creates the minimal diffusion distance needed for extremely rapid exchange.
And where would we find this in the body?
Classic examples are the lining of the kidney's Bowman's capsule, which facilitates immediate blood filtration, and also the lining of all your blood and lymphatic vessels, which we call the endothelium. In those places, gas or nutrient exchange needs to be basically instantaneous. These cells prioritize surface area and speed above everything else.
Okay, but what happens when we need a structure that prioritizes active work over just passive speed? That's where simple cuboidal cells come in.
Right. When a cell needs to be an active metabolic factory, a single thin sheet just isn't enough space for the machinery. Simple cuboidal cells are roughly square or hexagonal, and that shape provides a much greater cytoplasmic volume. Their nuclei are spherical and centrally located. This increased volume allows them to house the machinery needed for active processes, abundant mitochondria for energy, and lots of RER and Golgi complex for manufacturing and secretion. You find them lining the collecting tubules of the kidney and the follicles of the thyroid gland, where they are specialized for active secretion and controlled absorption.
And if we need the absolute maximum capacity for manufacturing or for highly specialized absorption, the high-volume factories.
Then we move to simple columnar. These are the tall cylindrical cells, the skyscraper cells of the epithelium. Their nuclei are typically situated down near the base. The benefit of this height is the massive volume of apical cytoplasm. This is where you can have specialized surface features, like the dense brush border of microvilli found on the intestinal lining. Those dramatically increase the absorption surface area.
So more surface area for grabbing nutrients.
A huge amount more. It also provides vast space for secretory granules. The small intestine, which requires high-level nutrient absorption, and many large exocrine ducts, which require complex secretion, are classic simple columnar domains.
Okay, so that's the simple single-layer types. Now we shift to the multilaminar, or stratified epithelium, and the function here immediately changes from exchange and capacity to pure, brutal protection.
That's the key trade-off. Stratified epithelia with 2 or more layers, they sacrifice the speed of diffusion that you find in simple layers in favor of mechanical resilience. They're specifically designed to protect against high stress, mechanical, or chemical abrasion. And crucially, they're categorized by the shape of the most superficial layer, because those are the cells that are taking the direct impact.
And the most common one, the real workhorse of multilayered tissue, is stratified squamous.
The standard form of protection. These cells undergo a transformation. They transition from cuboidal cells in that mitotic basal layer, and they flatten out into squamous cells at the surface. And here we have to distinguish between 2 different types based on their ultimate defense mechanism.
Right. The keratinized stratified squamous, which is our primary external shield.
Yeah.
Our skin.
Yes, the epidermis of the skin. In this type, the surface layers are not only flat, but they've undergone a process of programmed cell death, or apoptosis. They are dead, enucleated, and their cytoplasm has been completely replaced with a complex, tough protein and lipid mixture called keratin. This specialized arrangement forms a highly effective, tough, and water-resistant barrier, which protects against desiccation, physical trauma, and microbial invasion. You also find it in high-stress oral areas like the hard palate.
And then there's the non-keratinized type for internal wet surfaces.
Exactly. Non-keratinized stratified squamous tissue is found in areas that are subject to abrasion but are kept moist by internal secretions. This means they don't need to prevent water loss. Here, the surface cells remain alive and nucleated. Locations include the esophagus, the vagina, the cornea, and the soft lining of the mouth. It offers excellent protection against abrasion, but it would quickly dry out and fail if it were exposed to the air. The difference between the two just underscores the level of specialization needed for dry versus moist protection.
So moving beyond those standard stacks and shapes, we get to the true architectural marvels. Let's start with the pseudo-stratified epithelium. The name itself suggests some confusion, but the structure is actually quite clever.
It is a brilliant design. It appears multi-layered because the nuclei are scattered at different heights, and that leads to the pseudostratification. However, the key is that every single cell, regardless of how tall it is, makes contact with the basal lamina. It's almost always columnar, and its specialization usually involves modifications to its surface.
And where's its key functional location in the body?
The major airways, the trachea and the large bronchi. Here, it is typically ciliated. The cilia act as these tiny hair-like oars, and they move a layer of mucus in coordinated sweeping waves. This is the mucociliary escalator. It's vital for clearing trapped dust and pathogens out of the respiratory passages.
So it's a self-cleaning system.
A self-cleaning system. You also find a non-ciliated version in the male epididymis, where the cells have something called stereocilia, which are actually just exceptionally long microvilli designed for absorption and helping to move sperm along.
Okay, next up, transitional epithelium or urothelium. This is the one that physically changes its shape.
This is a highly specialized lining of the urinary tract, and it's designed to accommodate massive changes in volume. It lines the organs from the kidney calyces all the way down to the urethra. When the bladder is empty, the tissue layers are thick and the surface cells are large, often cuboidal or even pear-shaped. But when the bladder fills and stretches, those same cells flatten out dramatically. They become thin and plaque-like, and this increases the overall surface area of the lining.
And what protects the tissue from the high osmotic pressure and, frankly, the corrosiveness of the urine itself?
The structural ingenuity is all in the apical surface of those cells. They have a remarkably thick, specialized membrane that contains these rigid, proteinaceous plaques. These plaques are highly resistant to the hypertonic, high salt concentration of urine, and that prevents leakage and cellular damage. The urothelium is really a masterpiece of adaptive lining designed for chemical resistance under extreme mechanical stress.
We also need to talk a little more about those specialized cellular functions, like the ciliated epithelium we just mentioned.
Yes, we find ciliated epithelium not just in the respiratory tract, but also lining the oviducts. In the oviducts, the coordinated wave motion of the cilia helps to propel the ova toward the uterus. The ability to generate this coordinated movement is the key functional specialization of these cells.
And what about specialized receptor function? The source calls them epithelial sensoria.
These are epithelial cells that have adapted to detect external stimuli. They are found in our special sensory organs, the olfactory epithelium for smell, the taste buds, and specialized cells in the inner ear for audition. These receptor cells communicate directly with underlying nerve terminals. They act as the interface between the body and the environment, taking on a role that is far beyond simple protection or absorption.
And finally, one of the most mechanically fascinating specialized cell types, the myoepithelial cells. So these are epithelial cells that act like muscle cells.
This is a perfect example of functional evolution. Myoepithelial cells are contractile. They contain significant amounts of actin and myosin filaments, the very same proteins that drive muscle function. They are sometimes called basket cells because of their stellate or star-shaped morphology. They surround the secretory end pieces and ducts of glands like the sweat, mammary, and salivary glands.
So their contraction literally squeezes the glands to release their products.
Exactly. Their entire job is to contract and forcibly expel secretory products into the duct system. This assists in the rapid release of things like milk or sweat. What's truly unique about them is their origin. Despite having muscle proteins, they are derived from ectoderm or endoderm, which reinforces their identity as specialized epithelial cells that have acquired this contractile function.
Right, so glands are the body's factories dedicated to manufacturing and exporting chemicals, and as you said, they are formed exclusively by these specialized epithelial cells. This transition from a protective layer to a productive organ seems key.
It is. Glands form during embryological development when epithelial cells proliferate and then invade the underlying connective tissue. And their entire classification system hinges on one simple thing: whether they maintain or abandon their connection to the original surface epithelium via a duct.
Okay, so let's start with the one that keeps its connection, the exocrine glands.
Exocrine glands retain a patent duct system that leads back to the surface epithelium. Their secretions, whether that's sweat, saliva, milk, or digestive enzymes, exit onto an external surface, like your skin, or an internal free surface, like the gut lumen. Examples are incredibly common: salivary glands, sweat glands, and the exocrine component of the pancreas.
And then we have the cells that cut the cord entirely, the endocrine glands.
Endocrine glands are ductless. They lose all connection to the surface epithelium during development. Their secretions, which we call hormones, are released into the adjacent interstitial fluid, where they immediately enter the highly vascularized bloodstream or lymphatic vessels to travel to distant target organs. Classic examples are the thyroid, the adrenal glands, and the pituitary gland. This ductless design is absolutely critical for global systemic communication.
And we also see cells that don't need a full gland structure to act locally on their neighbors.
Indeed, we have paracrine and neuroendocrine cells. Paracrine cells are scattered within the epithelium or connective tissue, and they release their messengers to act only on neighboring cells, providing very local regulation. Neuroendocrine cells act as an interface, either signaling nerve endings or responding to neural stimuli. They are essential for processes like local blood flow control, nutrient sensing in the gut, and overall digestive regulation. They often package peptide hormones and neurotransmitter-like substances in these dense core granules for a very regulated release.
So understanding how a glandular cell releases its product is crucial. It dictates the energy cost, the type of product, and even the longevity of the cell itself. The source mentions 3 distinct mechanisms.
Right, 3 distinct structural and functional mechanisms for release.
Let's start with the most efficient and common way where the cell survives completely intact, merocrine secretion.
Merocrine secretion is essentially exocytosis. The secretory vesicles, which are synthesized by the ER and then processed by the Golgi complex, the cell's manufacturing and packaging plant, they migrate to the apical plasma membrane. They fuse with that membrane and release their contents outside the cell without any loss of cellular material at all.
So it's a clean, renewable process.
It's clean, but it is highly energy intensive. It requires significant ATP and calcium. But it is the metabolically superior way to produce large volumes of protein and peptide secretions. Simple sweat glands and the pancreatic acinar cells, which pump out massive amounts of digestive enzymes, rely exclusively on merocrine release.
Okay, then we have apocrine secretion where the cell sacrifices a small part of itself.
In apocrine secretion, the product accumulates near the apex of the cell, and then a small portion of that apical cytoplasm, encased in a membrane, is pinched off and released as part of the secretion. This involves a partial loss of the cell membrane and some cytoplasm. The classic primary example here is the release of the lipid component of milk from the mammary glands. This method is used when the product, like fat droplets, can't be easily packaged into small secretory vesicles.
And the most extreme method, holocrine secretion, which is basically cellular self-sacrifice.
It is. Holocrine secretion involves the entire cell accumulating the secretory product until it degenerates and ruptures, releasing its entire contents, cell debris, lipids, proteins, everything, as the secretion itself. Since the cell is entirely destroyed in the process, the gland must have a high mitotic rate to continuously replace the dead cells from the basal layer. The sebaceous glands, which secrete the oily sebum onto your skin and hair, are the canonical example. The resulting sebum is literally the broken down cellular components.
So it's a high-yield but completely destructive process.
Exactly, used primarily for lipid-rich secretions.
The structural organization of these glands also reflects the scale of their operation, right? From simple unicellular structures to these massive compound organs.
Indeed. The simplest form is the unicellular gland, where a single cell acts as its own factory. The goblet cell is the perfect example, scattered throughout the respiratory and intestinal tracts, specialized only to secrete mucin. Most glands, however, are multicellular.
And these multicellular glands are classified first by the complexity of their duct system. So what characterizes the simple glands?
Simple glands possess unbranched ducts, or sometimes no duct at all. They come in various shapes of secretory end pieces.
Yeah.
You can have simple tubular, which are straight like those in the intestine, simple branched tubular like gastric glands, simple coiled tubular like sweat glands, and simple acinar or alveolar, which have rounded end pieces like urethral glands. These typically handle smaller, localized secretory loads.
And for massive scale production for huge volumes, we need the compound glands.
Compound glands feature highly branched duct systems, and this architecture allows a single organ to serve numerous widely distributed secretory units. This dramatically increases the potential output volume. They are classified as compound tubular, compound acinar-alveolar, or a combined type, compound tubulo-acinar. Major organs like the pancreas and the salivary glands utilize this complexity.
And within those compound glands, we also classify by the type of product. Serous versus mucus. What's the difference there?
Serous glands produce a watery, often protein and enzyme-rich secretion. Because of their intense protein synthesis activity, their cells are structurally packed with rough endoplasmic reticulum, RER, and a large Golgi complex. This causes them to stain intensely basophilic, that's a blue or purple color in standard histology. The exocrine pancreas, which is responsible for producing massive amounts of digestive enzymes, is a classic serous gland.
And conversely, the mucus glands produce the viscous lubricant, mucin.
Right. Mucus glands secrete mucin, which is a highly glycosylated, viscous protein secretion. During the slide preparation process, the mucin granules are typically washed out by solvents, and this causes the cells to appear pale, clear, or vacuolated. Their nuclei are usually flattened and pushed toward the base of the cell. Goblet cells are unicellular mucus glands, and many minor salivary glands are purely mucus.
And what if a gland needs to produce both types of secretion at the same time?
Then you have what's called a mixed gland, or a seromucus gland. These contain both serous and mucus secretory units within the same organ structure. They're often arranged with the serous units forming a crescent, which we call a serous demilune, that caps the mucus end piece. the submandibular and sublingual salivary glands. And the terminology here is often confused, but it really just relates to the power of visualization you're using. The term basement membrane, BM, is what we see under a light microscope. It's often highlighted by staining regions like POS because of its carbohydrate richness. It just appears as a continuous acellular layer.
But the true detail, the engineering complexity, requires the magnification of an electron microscope.
That's when we can analyze the basal lamina, BL. The BL itself is composed of two primary thin layers. First, you have the lamina densa, which is the electron-dense layer. This is the structural core, composed predominantly of type IV collagen, which forms a resilient sheet-like network. And second, there's the lamina lucida, the electron-lucent layer, which contains key adhesive glycoproteins like laminin and specialized proteoglycans. While the specific nanometer thickness, 80 millimeter versus 40 millimeter, is a microscopic detail, the takeaway is that the entire complex is exceptionally thin, often only 120 nanometers, to facilitate that efficient diffusion we talked about.
And this critical foundation isn't only found beneath epithelia, which is interesting.
No, which really underscores its importance as a universal biological structure. The BL acts as an enveloping sheath, surrounding muscle cells, myocytes, fat cells, adipocytes, and Schwann cells in the peripheral nervous system. It connects them all to the larger extracellular environment and provides a supportive framework for their function and maintenance.
So let's talk functions. The BL is far more than just inert scaffolding. The source says it's a dynamic, signaling and organizing layer that is critical for health.
It is. Let's look at its four essential roles.
First, the obvious one, physical support and anchorage.
It provides the firm mechanical attachment site for the cells. Epithelial cells anchor themselves to the BL via specialized structures called hemidesmosomes and anchoring filaments. This secure attachment is absolutely essential for maintaining cell polarity, making sure the cell knows which side is the apical surface, the free side, and which is the basal surface, the attached side. And that polarity is fundamental to function, particularly for secretion and absorption.
Second, its astonishing job as a filter.
This is perhaps its most specialized function, and you see it most prominently in the kidney glomerulus. The BL there acts as a highly selective barrier, controlling the passage of molecules based on both size and charge. The negative charges carried by the proteoglycans in the BL repel negatively charged proteins in the blood, effectively preventing massive plasma proteins like albumin from escaping into the urine. This dual filtering mechanism ensures that filtration is both precise and controlled.
Third, scaffolding. It acts as a biological blueprint for repair.
This function is critical for tissue survival. The BL acts as essential scaffolding during wound healing and regeneration. If an epithelial sheet is destroyed, the remaining BL provides the precise molecular pathway. It guides the migration of new cells, and it guides the outgrowth of axons in the nervous system to reestablish the correct tissue structure. If this BL structure is lost or damaged beyond repair, the resulting healing is often disorganized, and that leads to scarring and functional deficits. In a way, it acts as the tissue's memory.
And fourth, regulation. The BL is constantly communicating with the cells attached to it.
Absolutely. The macromolecules within the BL laminin, type 5e collagen, heparan sulfate, they are active signaling ligands. They interact with cell surface receptors, notably integrins, and they influence cell polarity, drive differentiation, promote survival, and even regulate gene expression. The BL is constantly telling the cell, this is where you are, this is what you should be doing, and this is how you must behave to maintain your identity.
The insight here is really clear. Because the BL is so foundational, tiny defects in its tiny components lead directly to systemic and often catastrophic diseases.
That's the so what moment. When the molecular integrity of that structure fails, the filtering or the anchoring mechanisms fail with it. Take Goodpasture syndrome. This is a severe autoimmune condition where the body's own immune system targets the critical structural protein, type 5e collagen, specifically the alpha-3 chain in the BL of the kidneys and lungs. When that foundation is destroyed, you lose the ability to filter blood, and that results in rapid renal failure and pulmonary hemorrhage.
And we see a similar outcome from a genetic failure of the exact same protein.
That's Alport syndrome. It's an inherited form of renal failure caused by congenital defects in the type 5 collagen gene. The failure to construct a stable BL from birth means the filtration mechanism is compromised from the very start, leading to progressive kidney damage.
And finally, the connection to mechanical failure. The blistering diseases.
The pemphigoid diseases, like bullous pemphigoid, they vividly illustrate the mechanical role of the BL. These are severe autoimmune diseases where the body produces antibodies that attack key adhesion proteins within the BL, like BPA2 or type C collagen. When those adhesion points, those molecular rivets, are destroyed, the epidermis, the epithelium, loses its secure connection to the underlying dermis, the connective tissue. This results in the separation of the two layers and the formation of large, painful, fluid-filled blisters. It demonstrates that without those precise molecular linkages, the body literally tears itself apart under normal stress.
Okay, so let's shift gears now. We move from the highly cellular, tightly bound epithelial layers that rely on the underlying tissue for survival to that critical support system itself, connective tissue. And the defining characteristic here is that the tissue is defined not by the cells, but by the space between them.
That's the fundamental architectural reversal. Connective tissue, or CT, is characterized by a predominance of intercellular extracellular matrix material, or ECM, and it's the resident cells themselves that synthesize and maintain this matrix. So instead of dense sheets of cells, you have widely scattered cells that are suspended within a complex, often vast matrix.
So the components of that ECM, the matrix, must determine the tissue's properties. What makes up the ECM?
The ECM has two main components. You have fibers, which include the strong collagen, the flexible elastic fibers, and the delicate reticular fibers. And then you have the amorphous ground substance, which is a viscous gel-like solution in which those fibers are embedded. The type, the ratio, and the organization of these components will dictate whether the CT is tough, like a tendon, flexible, like skin, or specialized for storage, like fat.
What are the overarching functions of CT in the body? It seems to do a little bit of everything.
It provides the vital supporting roles. It physically supports and connects all other tissues, providing the mechanical framework, the scaffold for all the organs. It gives tissues their tensile strength and their structural integrity. And crucially, it facilitates transport and defense. Since it is highly vascularized, it carries blood vessels and nerve endings to feed the avascular epithelia, and it provides the migratory path for mobile immune cells to patrol the body.
And since it's so diverse, how is it categorized?
We broadly divide CT into ordinary or general CT, which includes loose and dense tissues, and specialized CT. Specialized CT includes structures whose matrix or cellular composition is highly modified, things like cartilage, bone, blood, adipose tissue, and hematopoietic tissues.
Okay, so let's meet the local population, the architects and builders of this matrix, the fixed residents.
The fibroblasts are the most critical fixed cells. They are the ubiquitous architects, and they're responsible for continuously synthesizing and maintaining all the components of the ECM, all the fibers and all the ground substance. When they are active, as they are during growth or repair, they are large, and they possess abundant RER and Golgi, which reflects their high output of protein, the fibers, for export.
And what happens when they slow down, when the building is done?
When they're quiescent or inactive, they become fibrocytes. They're smaller, spindle-shaped, and they contain much less cytoplasmic machinery. But fibroblasts are vital in repair. When tissue is injured, they become hyperactive, they proliferate rapidly, and they lay down new collagen to repair the gap. If this activity is excessive or disorganized, it leads to fibrosis or the formation of scars.
Next, the dedicated energy stores. Adipocytes, or fat cells, which are derived from fibroblasts.
Adipocytes store energy highly efficiently as triglycerides, and they are essential not just for energy reserves, but also for insulation and for shock absorption. And we have to distinguish between the two primary types based on their structure and their function.
Let's start with A, white adipocytes or white fat. This is the ubiquitous energy store.
White fat cells are unilobular. That means they contain a single massive lipid droplet that fills nearly the entire cell. This droplet pushes the nucleus and the thin rim of cytoplasm to the extreme periphery of the cell, and that creates the classic signet ring appearance in histology. Functionally, beyond energy storage and insulation, white adipose tissue is a critical endocrine organ. It secretes hormones like leptin, which regulates satiety, and adiponectin, which influences overall metabolism and insulin sensitivity.
Okay, now B, brown adipocytes or brown fat, PAT. This specializes in heat generation. And here's where we get that great aha moment of biological mechanism.
Right. Brown fat is multilocular. It contains multiple smaller lipid droplets, and it has a central nucleus. It has a very high concentration of mitochondria, which gives the tissue its characteristic brown color due to the high concentration of cytochrome oxidase in those mitochondria. The key function here is non-shivering thermogenesis. This is achieved through a protein that is unique to these mitochondria, and it's called uncoupling protein 1, UCP1.
Uncoupling protein. So what is it uncoupling?
Normally, mitochondria use the proton gradient that they generate across their inner membrane to power an enzyme called ATP synthase, which creates the energy molecule ATP. UCP1 acts as a short circuit. Instead of funneling that proton gradient through ATP synthase, UCP1 allows the protons to leak back across the membrane. This dissipates the electrochemical energy, not as chemical energy, not as ATP, but directly as heat.
Wow. So it just bypasses energy production to make pure heat.
Pure heat. This highly efficient heat generation is vital for newborns and for cold-adapted adults to maintain their core body temperature. It is a stunning example of dedicating a complex cellular organelle, the mitochondrion, to a non-standard but life-saving metabolic purpose.
And finally, the reserve pool of cells.
Yeah.
The mesenchymal progenitor cells.
These are undifferentiated, embryonic-like cells that persist into adulthood. They're typically found surrounding blood vessels. They retain multipotency, which means they can differentiate into any of the CT cell types, fibroblasts, adipocytes, chondrocytes, or osteocytes, as long as they receive the correct biochemical signals. They are the body's resident repair and regeneration crew, providing the cellular substrate for forming new tissues when needed.
Okay, that covers the fixed residents. Now for the wandering nomads, the mobile units.
Right. The CT matrix is the main highway for the body's defense and repair forces. These wandering cells are derived from the bone marrow, and they travel into the CT from the blood to patrol and respond to injury.
And leading the charge are the chief phagocytes, the macrophages.
Macrophages, which are known as histiocytes once they enter the CT, are large, highly motile phagocytic cells derived from blood monocytes. Their role is massive. They are the vacuum cleaners of the tissue. They engulf foreign material, cellular debris, dying cells, pathogens, you name it. They are essential in initiating immune responses and clearing up inflammation. And when they confront debris that's too large to handle individually, multiple macrophages can actually fuse together, forming giant multinucleated cells. It demonstrates their adaptive military strategy.
Next, the crucial inflammatory cells, mast cells. The source calls them the hidden alarm system.
That's a great description. Mast cells are ovoid cells that reside near blood vessels and nerves throughout the connective tissue. Their defining characteristic is that they are loaded with cytoplasmic granules containing potent mediators of inflammation. These include histamine, heparin, which is an anticoagulant ECFA, which stands for eosinophil chemotactic factor of anaphylaxis, and NCF, or neutrophil chemotactic factor.
And what triggers the rapid release of these potent chemicals? What sets off the alarm?
They are critical in allergic reactions and anaphylaxis. When the surface of a mast cell binds specific IgE antibodies, which are often triggered by an allergen, it initiates a rapid degranulation. The histamine causes immediate vasodilation and increased vascular permeability. That leads to the swelling, redness, and fluid leakage that is characteristic of inflammation. And the chemotactic factors, the ECFA and NCF, they serve to attract other immune cells, like eosinophils and neutrophils, to the site of injury or allergic reaction. They mobilize the wider defense force.
And the other key immune components we find patrolling the CT space.
We also find lymphocytes, T and B cells. These are small, numerous immune cells that constantly migrate between the blood, the CT, and the lymphatic system, and they provide targeted immunity. B cells are particularly important in the CT because when they're activated, they differentiate into plasma cells. And plasma cells are specialized antibody factories. You can recognize them by their eccentric nucleus and their massive Golgi apparatus for packaging antibodies. They are essential for long-term humoral immunity. And finally, the granulocytes, neutrophils, eosinophils, and basophils, they emigrate rapidly from the blood into the CT during acute inflammation, and they form the first highly lethal wave of defense, particularly against bacteria, for neutrophils, and parasites, for eosinophils.
Okay, we've established the cells. Now let's look at the actual materials they produce. The ECM, the non-cellular component, composed of fibrous proteins and ground substance. This is what provides the structure and the signaling medium.
The ECM is the scaffolding, it's the rebar, and it's the glue. Its composition provides the mechanical support determining tensile strength and elasticity, and it manages hydration, diffusion, and cell signaling pathways. The specific function of any connective tissue is fundamentally a reflection of the ratio and the type of fibers it contains.
Let's talk about the champion of strength, collagen. It's the most abundant protein in the body, giving tissues their incredible tensile strength. We mentioned it's a family of over 30 types, but the structural core remains that triple helix.
The triple helix, formed by 3 alpha chains twisted together, is the foundation of its strength. These are then polymerized into larger structures, forming fibrils and eventually the thick fibers we see under the microscope, which often display a distinct banding pattern. This synthesis and modification process is complex. It requires specific enzymes and cofactors, which makes it metabolically vulnerable to certain deficiencies.
Okay, let's break down the functions of the critical types. Let's give them an identity.
Type I collagen is the industrial strength material. It forms thick, powerful fibers that provide massive tensile strength. You can think of it as a biological steel cable. It dominates the dermis of the skin, bone, where it provides the organo-framework tendons and ligaments. It is responsible for resisting pulling forces.
Type 2 seems specialized more for cushion and pressure resistance.
Type 2 collagen is the pressure resistor. It forms fine fibrils, but it does not aggregate into thick fibers. It is restricted almost exclusively to hyaline and elastic cartilage. There, its fine interwoven meshwork allows the tissue to resist intermittent pressure and compression, functioning like a resilient shock absorber in your joints.
And type 3, the delicate framework.
Type 3 collagen forms fine, branching fibers that we call reticular fibers. They are delicate and they create a soft, supportive mesh or stroma. A key distinguishing feature is that they are argyrophilic. They stain black with silver salts, which makes them very easy to identify in histology. They provide the essential structural support for highly cellular, soft organs like the lymph nodes, the spleen, the liver, and the bone marrow. They hold the cellular components in a supportive sling.
And type 5 is our foundation layer, the sheet former. We talked about this with the basal lamina.
Exactly. Type 5 collagen is functionally unique because it does not form fibers. Instead, it polymerizes into sheet-like networks, making it the essential, resilient component of the basal lamina. It creates that pliable, permeable foundation upon which all epithelial cells, endothelial cells, and muscle cells rest. The source also notes type V collagen, which associates with type I in specialized tissues like the cornea, and tacti-6 collagen, which forms small beaded filaments that are important in cartilage.
We have to revisit the clinical connection here, specifically with scurvy. It so beautifully illustrates the molecular necessity of vitamin C for collagen.
Scurvy, which is caused by a vitamin C deficiency, is a failure of molecular synthesis. Vitamin C is a necessary cofactor for the hydroxylation of proline and lysine amino acids within the collagen molecule. Without proper hydroxylation, the 3 alpha chains cannot form a stable triple helix. The resulting unstable, fragile collagen leads to symptoms like fragile blood vessels, so you get bleeding gums, poor wound healing, and musculoskeletal pain. It demonstrates that the health of the entire connective tissue system relies on one tiny dietary requirement.
So if collagen provides the strength, elastic fibers provide the flexibility and the resilience, the ability to snap back to the original shape.
Elastic fibers are composed primarily of elastin, which is an amorphous protein rich in glycine and proline, and they have associated fibrillins. Elastin is unique because of its specialized crosslinks, desmosine and isodesmosine, which allow the fibers to stretch up to 150% of their resting length and then passively recoil.
And they must be crucial in organs that have to constantly change their volume or shape.
Absolutely. They are abundant in the lungs, allowing for recoil during exhalation, in the skin, maintaining elasticity, in elastic cartilage, and critically, in the walls of large pulsating arteries like the aorta. There, they dampen the pressure wave and allow the vessel to snap back after each heartbeat.
And there's a genetic vulnerability associated with their structural components.
The best-known example is Marfan syndrome. This is a genetic disorder caused by a defect in fibrillin 1, which is one of the proteins that forms the elastic microfibrils that guide elastin deposition. Without stable fibrillin 1, the elastic fibers are unstable and fragmented. This leads to severe issues in tissues that are under constant stress, most dangerously the structural weakening of the elastic aorta, making it prone to dissection and rupture, along with skeletal and ocular issues.
Okay, so we shift now from the reinforcement rods, the fibers, to the surrounding hydraulic gelatin, the ground substance. This is the amorphous, viscous material filling the space between cells and fibers, and it's essential for hydration and diffusion.
The ground substance is the physical environment in which all the CT cells live and through which all nutrients and wastes must travel. It's rich in water and inorganic salts, but its critical structural components are these large macromolecules, glycosaminoglycans, or JAGs, proteoglycans, and adhesive glycoproteins.
The JAGs sound like they're the core of the tissue's hydration capacity.
They are. GAGs are long, unbranched polysaccharide chains. Their functional superpower comes from the fact that they are highly anionic. They possess numerous negative charges from sulfate and carboxyl groups. Because of this high negative charge, they act like molecular sponges. They strongly attract and bind vast quantities of water via osmotic pressure. This creates a turgid, hydrated, gel-like medium that is highly resistant to compression. This is crucial for your joints and other soft tissues. Key examples include chondroitin sulfate, dermatin sulfate, and keratin sulfate.
And one JAG is unique and absolutely massive: hyaluronan.
Hyaluronan, or hyaluronic acid, is an exception among the JAGs. It is exceptionally large, it's non-sulfated, and it does not covalently link to a protein core. It exists as this massive free molecule in the matrix. Its primary role is lubrication and space filling. It provides the extremely viscous component of synovial fluid in your joints and the vitreous body of your eye. It organizes the water to create a loose protective scaffold.
And how do the other GAGs combine to create even larger functional structures?
They form proteoglycans. A proteoglycan consists of multiple sulfated DAG chains that are covalently linked to a central protein core. These individual proteoglycans can then non-covalently link to a central hyaluronan backbone, and that forms enormous structures called proteoglycan aggregates like aggrecan, which is found in cartilage. These aggregates reinforce the matrix, they regulate the diffusion of molecules, and they dramatically increase the viscosity and the compressive resistance of the tissue.
Okay, and the last component, the adhesive glycoproteins.
These specialized molecules are the biological superglues. They mediate the critical connection between the cell surface and the ECM components, and they essentially regulate cell communication, movement, and structural integrity.
Starting with fibronectin, the all-purpose linker.
Fibronectin is a large glycoprotein dimer. It is remarkably versatile because it contains specific binding domains for almost every component: for collagen, for heparin, and for the cell's surface receptors called integrins. It acts as a bridge, connecting the cell's internal cytoskeleton to the external matrix. This is critical for processes where cells must move or change shape, such as guiding cell migration during embryonic development or during wound repair. It provides a physical roadmap through the matrix.
And laminin, which you mentioned, is restricted entirely to that foundational basal amina.
Laminin is a large, cross-shaped glycoprotein found exclusively in the basal lamina. It is the core molecular anchor mediating the adhesion of epithelial, muscle, and nerve cells to the underlying BL. It binds to type 4 DAC collagen, heparin sulfate, and other proteins, creating the secure and stable adhesion that is necessary for cell survival and polarity.
And finally, kinesin, which the source calls the transient molecule of remodeling.
Tenascin is a large subunit glycoprotein that is mostly absent in stable adult connective tissue. Its presence is highly associated with dynamic processes, developing tissues, aggressive tumors, and active wound healing sites. It appears to play a role in regulating the matrix and guiding cell migration during rapid tissue remodeling, which demonstrates that the ECM composition isn't static, but changes rapidly in response to environmental cues.
Okay, so by varying the ratios of all these components, the cells, the fibers, the ground substance, we create these distinct functional classes of general connective tissue, each tailored for specific mechanical needs.
The classification is entirely driven by the density and the orientation of the collagen fibers, because that's what provides the mechanical specialization.
Let's start with loose or areolar connective tissue. This sounds like the most widespread type, the packing material of the body.
It is the least specialized and the most flexible. It is characterized by a loosely woven arrangement of all the fiber types: collagen, elastic, reticular, embedded in abundant ground substance. It is highly cellular and highly vascularized, which makes it look a bit like a sponge under the microscope.
Functionally, it sounds like the tissue's central hub.
It is. Loose CT fills the spaces beneath all epithelia, forming the lamina propria of mucous membranes. It surrounds vessels and nerves, and it connects the dermis to the underlying muscle. Its primary functions are binding structures together, filling space, and acting as the primary site for inflammation and immune reactions because of its high population of pneumatic immune cells and its vast vascular network.
So moving to the dense tissues, we dramatically increase the fiber content. Dense irregular CT is built for omnidirectional mechanical strength.
This tissue is mechanically robust. It's defined by dense bundles of type I collagen fibers that are interwoven and oriented in various directions. That's the irregular arrangement. It contains fewer cells and much less ground substance than loose CT. The multidirectional fiber orientation provides massive strength and resistance to tension and stress that's applied from multiple planes simultaneously. You find it in the deep dermis of the skin and forming the tough protective capsules of internal organs like the kidney, the testes, and the spleen.
And then there's dense regular connective tissue. This is the ultimate structural specialization, built for overwhelming strength but in only a single direction.
This is the unidirectional champion. In dense regular CT, the collagen bundles are highly organized, packed tightly, and oriented strictly parallel to the direction of maximum tensile stress. This maximizes its resistance along that single axis. It contains minimal matrix, and the cells, the fibrocytes or tendonocytes, are aligned these precise rows between the parallel fiber bundles. This immense directed strength is required for structures like tendons, which connect muscle to bone, and ligaments, which connect bone to bone.
And finally, we should revisit those specialized types that use the CT framework for dedicated non-mechanical functions.
Right. Adipose tissue is the specialized CT composed almost entirely of adipocytes organized into lobes separated by septa. As we discussed, it's a highly vascularized energy store, an insulator, and an endocrine regulator.
Next, reticular tissue, the delicate meshwork.
Reticular tissue is defined by a delicate meshwork, or reticulum, exclusively of type 3 collagen fibers. Its function is to provide the soft, supportive stroma, the internal framework that physically supports the massive cellular populations of hematopoietic organs, such as the bone marrow, the spleen, and the lymph nodes. It allows those cells to organize and operate efficiently.
And mucoid tissue, the flexible filler.
Mucoid tissue is unique because of its large amounts of highly hydrated ground substance, primarily hyaluronic acid, and its widely separated fine collagen fibers. This composition provides flexible, resilient support. The most classic example is Wharton's jelly, which surrounds and protects the blood vessels within the umbilical cord, giving the cord its necessary flexibility and turgidity.
We often talk about biological membranes, but it's important to remember they're actually complex sheets of tissue. Always an epithelial layer plus the underlying connective tissue.
These composite structures perform large-scale organizational roles, and they're defined by their location and their environment. The two major types are the mucosa and the serosa.
Let's detail the mucosa, or mucous membrane.
Mucous membranes line all of the passages that communicate with the external environment, so the digestive, respiratory, and urogenital tracts. They're typically made of 3 layers: the specialized epithelium, which can vary widely in type based on a location, the underlying lamina propria, which is loose connective tissue for support and defense, and often a third thin sheet of smooth muscle called the muscularis mucosae. Their function is complex: lubrication, absorption, and protection. And this is achieved by keeping the surface moist with secreted mucus.
And the serosa, which lines the sealed internal cavities of the body.
Separous membranes line the sealed cavities, the peritoneal, pleural, and pericardial, and they cover the organs within them. Structurally, they are a simple layer of squamous mesothelial cells that overlie a thin layer of CT. Their key job is lubrication. They achieve this by secreting a watery, protein-containing serous fluid that minimizes friction, and this allows organs like the heart and the lungs to glide smoothly against the internal walls during movement.
So a critical insight here is that this highly specialized system is not rigid. The cells and tissues demonstrate this remarkable plasticity, capable of changing their identity, a process the source calls transdifferentiation or metaplasia.
It is a critical insight, yes.
Metaplasia sounds like the body choosing the lesser of two evils in response to some kind of chronic injury or stress.
That is the perfect way to synthesize it. Metaplasia is the reversible conversion of one mature differentiated cell type to another mature cell type, and it's always in response to chronic irritation. The conversion is adaptive. It allows the tissue to better withstand the ongoing stress, but it always comes at a cost or a trade-off.
Give us the specific examples that demonstrate this trade-off. What is the cost?
In the respiratory system, for instance, in a chronic smoker, the delicate, specialized, simple columnar ciliated epithelium in the trachea, which is built for sweeping away mucus and particles, is chronically irritated by toxins and heat. It shifts to a much tougher, multi-layered stratified squamous epithelium. Now, the tissue is far better at resisting mechanical abrasion and chemical toxicity, but the severe trade-off is the complete loss of the mucociliary escalator function. This leads to chronic coughing and particle retention.
So it gains protection but loses its primary function. And the gastrointestinal example with acid reflux.
That is Barrett's esophagus. Chronic acid reflux damages the protective, non-keratinized, stratified squamous epithelium that is normally found in the lower esophagus. In response, the tissue converts to a columnar secretory epithelium, which is a type that is better at secreting mucus and resisting the constant chemical insult of the stomach acid.
So adaptation comes with a significant risk.
It does. Metaplasia confirms the incredible plasticity of our tissues. It shows the cellular drive towards survival under duress. However, this adapted state is often premalignant. The conversion to a cell type not usually found in that specific location often carries a significantly higher risk of progressing toward malignancy or neoplasia. It makes it a functional, short-term win that carries a serious long-term risk.
So from the elegant, speedy simplicity of simple squamous cells built for maximum diffusion, all the way to the immense, organized tensile strength provided by type I collagen and a major tendon, we have covered the incredible architectural genius of your body's foundational tissues. The integration of just these two cell categories, epithelium as the boundary and connective tissue as the structural and metabolic engine, is responsible for protection, movement, filtration, communication, and overall mechanical integrity across all your systems.
We saw how the basal lamina serves as the tissue's molecular memory, guiding regeneration and setting cell polarity. We learned that the difference between simple healing and systemic disease often relies entirely on the successful synthesis of highly specialized matrix proteins like type 5 collagen, or the crucial activity of fibroblasts in synthesizing the ECM. So if we connect this back to the absolute beginning, the fact that all four major tissue types, epithelium, connective, muscle, and nervous, arise from just three primary embryonic germ layers, consider this final provocative thought. To what extent does the molecular composition of the initial basal lamina laid down during development determine and permanently lock in the entire functional fate and architectural identity of the tissues they support throughout the lifespan, pre-programming their strength, their weaknesses, and their potential for change? Something to contemplate as you consider the hidden architecture beneath your own skin.